be our next speaker.
Thank you. So today I'm going to talk about a new project that we've been working on in my lab, where we're able to take different forms of biomass and convert it into both gasoline, that is the same gasoline that you now use in your cars, as well as other petrochemicals. And so I'll show you the biological step in that route. So clearly the world is facing an energy crisis with the growing economies of many countries, including those in this region. We have tremendous pressures on our energy supply. So this is showing the growth in wealth per capita versus the growth in the amount of energy that's being used. And you can see where we are up here, and then countries like Korea have grown tremendously over the years. Here's China and India, so about 1 billion people each. And then there's another 2 to 3 billion people that are not on this curve altogether. And so this has led to two problems, both of which are well known. The first is the accumulation of greenhouse gases, which is affecting our climate change. The other is the forces on supply, which is causing costs to skyrocket. And so I think there are only two possible technologies that can address both the supply and the environmental issues, and those are advanced biofuels and hydrogen production. Hydrogen, of course, has additional problems with the infrastructure, but I think there are some interesting applications for synthetic biology in that arena, and many of which are part of the poster session. So today I'm going to talk about advanced biofuels, and I think in some of the previous sections we've already seen what some of the key problems are with the first generation biofuels, like ethanol and so on. So rather than outline those, I'm going to point out one key difference between the biocarbon economy and the existing petroleum economy that I think this work really addresses. And that is what I call the fungibility of carbon. So the last 150 years of chemistry and chemical engineering have made it such that you can get your carbon from natural gas, you can get carbon from oil, you can get carbon from coal, and by doing a variety of transformations, you can take that carbon and turn it into gasoline, into a liquid fuel, or you can convert it into commodity chemicals like ethylene and benzene and propylene and so on, which then add as a feedstock for the rest of the petrochemical industry. So it is these core molecules which then get turned into all of the things that we use out of petrochemicals. And so this is in stark contrast, I think, with biocarbon. Biocarbon is not fungible both in the inputs and the outputs. You can't go to an ethanol plant, a corn ethanol plant, and dump off wood chips instead. And you can't have an ethanol plant make ethylene instead of ethanol, right? We require a different organism, a different process entirely. So I think what you really have to address as you're solving the fuel problem is also how we're going to supply carbon into all of the variety of products in which we see out of this industry. So the Department of Energy in the United States recognized this problem, and they have a simple-to-use framework for how they go from biomass to products, meant to guide the research. I've simplified it a little bit here. The basic idea is very simple, what they're calling for. You start out with carbon that's locked in the form of a feedstock. So you might have sugars or starches, cellulose, which we've heard about and we'll hear more about, even maybe carbon dioxide and maybe even petroleum. And then what they imagine is a two-step process where the first step is a biological step where you use cells and genetic engineering to go in and reduce some of the functionality of the highly functionalized sugar and cellulose and so on. And so what you do is you have the cells make a building block molecule where if you pick the correct building block, this can then feed into existing catalytic processes by which you can build all of the fundamental things that then get turned into products. So you have to be very clever about what building block you pick, and it has to be consistent with the existing infrastructure. So here are some of the required properties for a chemical building block that you're going to have biology make. First, it has to be a high percent theoretical conversion from biomass. We heard James talk a little bit about this. So because you're talking about commodities and fuels, most of the carbon in your biomass has to turn into product for it to be economically viable. It has to be cheap and energy efficient to purify the product. It has to be functionalized so that you're not adding a lot of energy just to make the molecule be able to be converted into other things, but it can't be too functionalized so that it cokes up the catalyst and so on. It has to be something that can connect with an existing catalytic framework that has been demonstrated to be consistent with the scale of petrochemicals. Okay, so it can't be very expensive catalysts that you're working with. And then there are two requirements on the side of the biology. The first being that it has to be something that's portable between host organisms, because this is how you're going to be able to use different biomass feedstocks. And then it has to be something that's non-toxic to the host. And I think a variety of different chemicals have been suggested as being an important potential building block molecule, but one was overlooked that has all of these properties. And that is methyl halides. And methyl halides, methyl chloride, methyl bromide, and methyl iodide, have already been studied in the context of going from natural gas sources to liquid fuel as well as commodity chemicals. So it's been shown that you can take natural gas and then in a very expensive step, make the methyl halide. And then with catalysis, you can make fuels or alkanes, aromatics, benzene, toluene, xylene, different alcohols and ethers, and olefins. And these are huge markets because these are the fundamental chemicals that then feed into the rest of the chemical supply chain. And I'll give you one example that I think is particularly exciting. This is work where methyl bromide that had been obtained by converting natural gas is run through a catalyst, ZSM5. This is a very common catalyst. And then the product that comes off is liquid gasoline. And here you can see the fraction of the gasoline versus the chain length. It looks very much like gasoline. The distribution between aromatics and alkanes is also identical to gasoline. This is gasoline. It's not biogasoline or a substitute or a blendable component of gasoline. It's exactly gasoline. It's what you put in your car right now. Just to point out what type of catalysts are used in this process, if you're a chemical engineer, you'll be very familiar with zeolite-based catalysts. This is essentially volcanic rock. It's been used since the 70s or possibly earlier for most of the catalytic conversions that already occur in petroleum and petrochemical industries. They're essentially solid-state acids, and they have a unique pore structure. And based on the acidity in the pore structure, you can essentially convert any carbon input to any carbon output based on the choice of catalyst and temperature and pressure and so on. These are already used at a huge scale. Here's an example of a plant in Bintulu, Malaysia, which is in Borneo. This is run by Shell. And these are big reactors containing these catalysts that are being used for the conversion of natural gas to liquid fuel. Now, as it turns out, methyl halides are naturally produced in the environment. There are a bunch of biological sources that do this, and they had been largely, well, completely overlooked from the perspective of biotechnology. They had been studied almost exclusively from the perspective of environmental chemistry, where things like methyl chloride will escape up into the atmosphere, react with ozone, and are a major ozone depleter. And so people had gone in to look at plants and organisms that do this, including rice, right, which is a major agricultural crop, with the idea of creating plants that don't produce these molecules and therefore don't harm the environment. And so one of the genes that came out of one of these searches is called HOL1 from Arabidopsis, and that stands for harmless to the ozone layer 1. So it's a very simple process by which the organisms make methyl halides. It's a single enzyme. It's only 230 amino acids. It's monomeric. It uses It directly converts a halide ion, which is usually provided as a salt, takes a methyl group from S-adenosyl methionine, and in an ATP-dependent manner makes the methyl halide. So S-adenosyl methionine is a good precursor for a pathway. It's something that's present in all cell types. It is a metabolite that's second in abundance only to ATP. It's very common. And something that's interesting is that it's already been overproduced in different strains because it's used as a nutraceutical, in particular in the United States and Japan. So there's strains of yeast that make many tens of grams of S-adenosyl methionine, and it doesn't seem to harm the cells. So that really speaks to the possibility of metabolic engineering coming in and getting some improvements. So we're now in the synthetic biology era. So rather than starting with a single enzyme that we thought had this function, we used our target sequence and went into the sequence databases and looked for any gene that looked anything like it. And we came up with a list of about 90 enzymes. We computationally optimized them for expression in yeast and in bacteria, and then we shipped them off to get synthesized. And about 2 weeks later, they came back in the FedEx packet. We could throw them into E. coli and screen them very quickly because these molecules are coming off in the headspace gas, and you run it through a GCMS, and it's very trivial. So this is a complete screen for all of the organisms that make methyl halides. This is for the production of methyl iodide. They're colored by the type of organism that they are. So in green, we have plants, red, bacteria, purple, fungi, and there's even an archaea in here somewhere. This is the screen for methyl iodide. If you look more closely, our best organism is from swamp wort, which is something that likes to grow in brackish water. Pinot Noir grapes make this. Burkholderia is an organism that lives in the soil. We're all familiar with bok choy, I think. And then one came out of, I think, one of the JCBI's searches of the ocean diversity, and so you don't even know what organism it is, much less what the enzyme did, and those turned out to be our top 5. This library had an extraordinary amount of diversity in it. If you look at the average amino acid diversity between all of the 89 sequences, it comes out to about 28%, right? And many of these sequences went down to 18% identity with our original target. If we just look at our top 6 sequences, it only reduced that increased that identity up to 39%. Right, so even in our top sequences, we have an extraordinary amount of diversity that's still present in the library. We went in and screened all 89 for the production of methyl iodide, methyl bromide, and methyl chloride, which are valuable for different reasons. We found different interesting activity profiles, like Batis maritima is the best producer of all 3, whereas this organism only makes bromide. This one basically only makes methyl iodide. This makes bromide chloride, but not iodide, and so on. So there's a lot of functional information that we can start going into this library and try to dig out the relationship with sequence. Something that I find fascinating is that out of all of the enzymes that we screened, only 6% had no activity on all 3 ions. And that's remarkable because only one was annotated methyl halide transferase, and only about 50% were annotated methyl transferase, right? So we went really far out as far as the diversity that we included in our library. So again, S-adenosyl methionine is present in all cells, so this is something that we ought to be able to easily move between organisms. So we moved it into yeast, and one of the advantages of yeast that I want to point out here is that it has a vacuole, and into that vacuole, excess S-adenosyl methionine is dumped. Ions are concentrated there. So we included a tag on the methyl halide transferase gene that directed it to the vacuole, and we found an improvement in the activity when we did that. Something else that is interesting, again, going to James Lau's talk, is that if you look at the theoretical yield that you can obtain, 4 of the carbons in sugar can get converted to methyl halide. So that is equivalent to ethanol production in theory. So these are the yields. When we moved it into yeast, we get a very constant production rate that keeps going. In this case, we're doing shake flasks, so it's tedious to try to collect the gas as you go. Here is a production of methyl iodide. This is the natural producer. E. coli, we got some improvement. And when we moved it into yeast, both in glucose and sucrose, we get about 200 milligrams per liter per hour of methyl iodide production, which is pretty remarkable given that we haven't even started the metabolic engineering. That's a 12,000-fold increase over natural sources. Now, methyl iodide and all of the methyl halides are non-toxic to yeast. So you can go up to between 5 to 10 grams a liter before you start seeing toxicity issues. And remember, this is coming off as a gas. So if you do a very simple chemical engineering 101 calculation, even if the cells are making 50 grams per liter per hour of product, the steady state concentration in the liquid only be 2 grams. So even in this unrealistically heavy case, we're going to be well below where we see toxicity in the system. This ability to come off as a gas improves our ability to collect it. And here I'll show you a couple movies that demonstrates this. The fun parts. Here we go. Okay, so in this movie, a student has a flask of yeast that are producing methyl iodide, ignite it in the hood. That one's not working, unfortunately. This one is a similar experiment, but in this case, we're pumping in air to simulate what would be happening in a fermenter that's being aerated. And you'll see the product coming off. This is the natural production of this. And what you'll see is we'll ignite it, and it'll have a blowtorch type effect right about there. So this is what has been naturally produced here, and start blowing in air, and product is going to come up, right? And you'll notice there's quite a bit that actually gets forced out by this. So it's very simple. It's very easy to condense the methyl halides. You can put them through a water-cooled condenser, and it drips off. The boiling point is very favorable for the collection of the product. And it just keeps going. Okay, so that's for the production of methyl halides. In those cases, we're using glucose, we're using sucrose, looking at roughly what would happen if we were using sugarcane as a feedstock. We've heard all of the reasons why we need to move away from food-based biomass stocks. And I'll just point out one that I think is particularly visually effective. This is looking at either the consumption or the production of carbon in the world. So this is all of the gasoline that's currently being consumed, all of the diesel, and so on. And then here we're showing all of the carbon that is currently produced in the form of different agricultural products. Okay, so this bar here, for example, is all of the corn that's being produced in the world. This bar here is all of the rice that's being produced in the world. Both of those are about 15% of what we currently consume in the way of transportation fuels. So if we took every grain of rice, every kernel of corn, converted them to biomass, we'd be able to address about 30% of the problem. But if we move to non-food forms of biomass, it's been predicted that we have an enormous capacity to be able to do that, to cover our fuel needs. The problem, of course, is that carbon is locked up in the form of polymers in biomass. So if we take something like a grass, zoom in on that plant wall, we would find polymers where eventually you would get to sugars, but you would need some way to be able to liberate those sugars such that the yeast or the bacteria are able to eat it. And the problem really has been that this plant mass is so complicated that it becomes very hard to give organisms that don't naturally eat it the ability to eat it or to use purified enzymes to break it down. That's proven to be a very difficult task when you're using unprocessed biomass. So what we decided to do was to look at an organism that has the natural ability to eat biomass, and we found one that we felt was particularly interesting. It's a very unique organism. It just got reclassified. It was incorrectly classified for 20 years. It is a organism that was collected from a French garbage dump. It grows both anaerobically and aerobically, and it has the natural ability to eat biomass. And what's interesting is that a very high percent of what it eats comes out as acetate and ethanol. Okay? The problem with using this organism by itself is that it's extremely strongly inhibited by its own products. Okay? So it doesn't naturally grow to very high of a cell density. So what we decided to do was to create a co-culture between this bacterium and the yeast that we were using, where the idea was that the yeast would be able to convert the acetate and ethanol into product, and then in that process would actually relieve this product inhibition of the bacteria. And then this whole system, because of this negative feedback loop, could be more stable as you move into a bioreactor type scenario. So these are the first experiments that we did with this co-culture. What we're using here is carboxymethylcellulose, which is purchased from Sigma Genesis. In many ways, it's cheating because it's a soluble form of cellulose that your grandmother could break down. It's a very easy target, but it was a good one to test this out on. So here we're looking at the relative growth. This is from the perspective of the yeast. If we don't have the bacteria, it can't grow on CMC as a carbon source. If we add this bacteria, then the yeast is able to grow. Similarly, if we're looking at the bacteria, without the yeast, it grows up to a low cell density. With the yeast, it relieves that negative feedback loop and is now able to grow to a higher density. And so it's really, truly a symbiotic relationship. So here we're making methyl halides using the co-culture system. Again, we're using carboxymethylcellulase. Without the bacteria, we don't produce any of the product. With the bacteria, now we're producing methyl iodide from cellulose. So again, CMC is cheating a bit. What we wanted to do is to use this organism's intrinsic ability to break down real sources of biomass to then allow the yeast to grow on these materials. One of the key targets in the United States is known as switchgrass. It naturally grows in the Great Plain areas. It has advantages like it can be mowed rather than reseeded each year. It doesn't require fertilizer and so on. And so we got some of the switchgrass from the USDA. And just to stress that we literally take the material that we receive and mix it with a minimal media. Okay, so it's as rough of a media as you can imagine. There are other forms of biomass that are also relevant. Corn stover, which is the leftover bits of corn. Wood chips are important in the Northeast. Bagasse is a sugarcane derivative. And for the bagasse, we just left it alone. For the corn stover and wood chips, we put it in the blender, and this was the extent of our pretreatment. We were inspired by those Will It Blend YouTube. So this is what we get from all of the different sources of biomass. On CMC, we get about 2.5 grams per liter per day. Cane bagasse, it goes up to about 3.5. Corn stover is doing very well. We get less with respect to poplar and switchgrass, but it's still, a reasonable 0.5 gram per liter, which is a good place to start, I think. But I think this is really showing the idea that if you pick the right organism, it can then feed into all of these different sources of biomass rather than having to focus on just one. So again, I'll just conclude with a summary of our approach. What we're trying to do is work on a two-stage process where we use a biological fermentation to take different sources of biomass, whether it be sugars, starches, or different forms of cellulose. The cells then can convert that into a methyl halide, which is easily collected. This is run through existing catalytic systems where you can recover the halide But then using this catalysis, you can make gasoline, you can make benzene, toluene, xylene, ethylene, propylene, methanol, formaldehyde, or dimethyl ether, all at high yield. And these may sound boring, but they're key chemicals. If you're looking at going into the supply chain for the petrochemical industry, where again, this is a overly complicated DOE report, where they're outlining what happens to petroleum and natural gas in the supply structure. Petroleum gets turned into benzene, xylene, toluene, and butanes, get converted into products eventually. Natural gas goes to ethylene, so on. And so if you look at what biological methyl halides can feed into this process, they can actually fairly well span the entire petrochemical set of feedstocks, which then feed into everything that we consume. So with that, I would like to end by thanking the people that have been involved in this research. This has really been Travis Bayer's project. He has a poster out there. He's been helped by Dan, by Ethan, and by Karsten. Howard is also here. He has a poster on a separate project. And thank the various forms of funding. You'll see we're a very happy lab. And it's because an orangutan was taking a photograph. But we'd be very happy to have you join us. Thank you.
Do we have time for a few questions?
The catalyst, ZSM5, does that work at ambient temperatures?
No, it's not. So the catalyst is typically, it depends on what product you're making, between 3 to up to 600 degrees Celsius. Yeah.
So how much energy do you have to put into heating the catalyst in order to make to make fuel from your precursor? How much energy do you lose total? Is it a net gain or?
Yeah. So the process that's been developed in detail is actually methyl bromide to liquid fuel, and the cost of that step is, which is energy as part of that, is very favorable. One of the really exciting things about this research is that it's been impossible to make methyl iodide out of natural gas via synthetic chemistry. We're able to make a lot of methyl iodide. Because it's more reactive, that could reduce the temperature, which would reduce the energy costs, reduce coking, and so on. But again, it's already a process that's in existence that we're feeding into, and the energy is not that much of a problem.
You didn't say what the growth rate of this bug that you picked up is, and the question with this is, how long can you sustain a fermentation co-cultivating these two organisms?
So first, the growth rate. So this French bacterium, you've hit on a problem, actually. So this French bacterium grows very fast, but it's only willing to grow 35 hours a week. I knew I should do it. It's a doubling time of 4 hours, which is a reasonable start. It's not super speedy, but it's certainly fine. Yeah. So in this case, we're doing everything in shake flask still. So we don't have any data regarding fermenters yet.
Hi, Chris.
Yes.
Does your library give you any insight into the enzyme mechanism of the transferase?
That's been yes. We're just starting to even develop the tools that can handle this level of diversity and trying to pull out structure function. But you can do things like say, give me all the enzymes that only make methyl bromide, give me all the enzymes that only make methyl iodide, and you find a key set of residues. So
do any do fluorination too?
What's that?
Will any do the fluorination step? I know that.
So fluorination has an additional problem of desolvation. And so I think for these enzymes it's been tried and it does not fluorinate.